Well, hello, AP Bio students. This is Mrs. Shelton, and today we are going to be reviewing Unit 3. Unit 3, as I'm scrolling backwards here through time, Unit 3 is a big dog. Unit 3 is Cellular Energetics. This was Chapter 6, 7, and 8 of your course. I'm going to be going over just some of the learning objectives that are outlined through the AP Biology CED, the course exam and description. Now, with this particular chapter, really what we are addressing in this chapter is we are addressing enzymes, we are addressing energy, we are addressing photosynthesis, and we are addressing cellular respiration. So that is why I said this is a big dog. This is one that you guys probably feel a little insecure about. Good news is we went over it in detail chapter-wise. Also, you ended up having, for your final from first semester, you had to go back over it. So this should be your third time reviewing this material. But again, remember that with the format of the 2020 exam, since it's all FRQs, don't worry about the minutia of the details. Understand the whole big concepts is what you should be familiar with. So with this, as we take a look at our different topics, the first three topics are enzymes. Enzymes are going to be the easier part of what we address. And then we'll talk about cellular energy, understanding, remember, our laws of thermodynamics. And then we'll get into details because we do have to understand like chemiosmosis and some relationships with photosynthesis and just how photosynthesis evolved. Cellular respiration, we need to make sure that we are addressing. And then we also need to understand a little bit about fitness. And I'll give you some illustrative examples at the end there. But this is, again, chapter 6, 7, and 8 from your textbook. And we will go over some of these learning objectives and go over some of the main points. You may want to make sure you go over those practice tests too. And then also read through your review, your test prep review series book too on these, just to make sure that you're seeing lots of different ways that this content could be addressed and asked. So first, as far as the first learning objective that we need to be aware of with enzyme structure, super, super easy. So with this, remember enzymes, these are biological catalysts, they're made of protein, and it's their shape that determines their action. What we need to make sure that we are understanding is that with our enzymes, there is the active site. The active site is the region that will collide with the substrate. The substrate is the reactant that the enzyme is going to help catalyze the reaction for. So remember your substrate and active site, they match as far as shapes are concerned. So those are the terms that we need to make sure that we are understanding is our enzyme substrate and then also active site. Now, with enzymes, how they work, the main thing that we need to make sure that we are understanding is we're understanding that enzymes are catalysts. So what catalysts do is catalysts speed up the rate of a reaction. And remember, I drilled this into your head during the school year, is how they speed up the rate of reaction is they reduce the activation energy. So here's a graph that should be very familiar to you. And remember that if we have no enzyme, it requires a tremendous amount of activation energy. And with an enzyme, then we reduce the amount of activation energy required in order to initiate or speed up that particular reaction. Remember, in the end, delta G, the amount of free energy released or required for the reaction, does not change. The only thing that enzymes change is the lowering of the activation energy is what they're going to be doing. Now, the third concept that we need to be addressing is the environmental impacts on enzyme function. Main thing to make sure that we are understanding with enzymes is this term denaturation. Remember that enzymes being proteins, their shape is everything, their conformation. And we know that pH and temperature can denature that protein, denature proteins, and if it denatures the enzyme, then the active site changes shape and therefore it doesn't function. But there's some other things that we need to make sure that we are understanding in here. We need to make sure that we are understanding, again, denature. We're understanding the role of pH and temperature and some other environmental impacts. We also need to make sure that we're understanding some inhibitors and also allosteric regulation as well. It's in this particular content that they're expecting you to understand pH a little bit more and what that pH value is. But here is 4 different graphs that are helping you understand some of the environmental impacts or influences on enzyme action. So enzyme concentration, substrate concentration, temperature, and then also pH. Now, with enzyme concentration, if I add more enzymes increase the rate of reaction. If I have a constant amount of enzyme, but now I increase substrate concentration for a while, I'm going to increase that rate of reaction. But because enzymes stay constant, there is going to be this point of no increase in the rate of reaction because my enzymes are at their max capacity for catalyzing reactions. Now, down here, when I talk about temperature, we do need to understand with temperature, it's not just denaturing of the protein that is going to affect it. Remember that kinetic energy. Enzymes act because of the collision rates. And so if I'm increasing temperature, I'm increasing the rate of reaction because the collisions are happening at a faster rate until I get to a point where now temperature is now denaturing those enzymes, and now we get this sudden drop off in enzyme activity. And then also remember that pH oftentimes affects that secondary structure of proteins being those hydrogen bonds. And so what ends up happening is enzymes all have an ideal pH in which they are optimal. This was an example that we talked about, like the enzyme amylase in your mouth. Works perfect in your mouth at that pH. As soon as it hits a pH of 2 in your stomach, well, now it no longer functions. So every enzyme in our body has an optimal pH where that ideal shape or conformation is going to occur for the enzyme, so it can also increase its activity. Now, the last thing about enzymes for this content that we need to make sure that we're understanding is enzyme inhibition. And with this, we do have other inhibitors that can either act competitively. So inhibitor means it's going to stop or prevent the enzyme from working. And we either have competitive or non-competitive inhibitors. And if I'm talking about competitive inhibitor, this is a molecule that actually gets in the way of the active site, blocking the substrate and therefore stopping that catalyzation of this particular reaction. I have non-competitive inhibitors. Remember, I described these as the backstabbers. So they are not attaching to the active site, they're attaching somewhere else in the enzyme and altering the shape of that enzyme, so now the substrate doesn't fit into the active site. Now, this non-competitive inhibitor is very similar to this allosteric activator or an allosteric inhibitor. So remember that in feedback pathways, oftentimes an end product can actually act as an allosteric, as an inhibitor in order to stop the process. And we also have this production of some other allosteric inhibitors or activators in order to also control enzyme action too. Because remember, for every biochemical pathway, there is an enzyme that is actually catalyzing each individual step. Now, for 3.4, in 3.4, the content that we need to make sure that we are remembering here is mainly we need to be familiar with the second law of thermodynamics, which is entropy. Nature tends towards disorder, or an increase in entropy is more randomness. Well, what we have to also remember is that living things are highly ordered. In order to maintain this high level of order, we need a constant input of energy. And remember that living things do not defy the second law of thermodynamics because in this constant input of energy, we're actually increasing the entropy in the universe in that process. So with this, there's some terms that we need to make sure that we are reviewing and remembering. We need to be remembering in here a delta G. Now Gibbs free energy equation, it's going to be given to you on the equation sheet. You don't have to memorize it, but we do need to make sure that we are remembering just some of these terms when we talk about exergonic reactions. So our exergonic reactions that are giving off energy, like the hydrolysis of ATP to ADP. This is an exergonic, a negative delta G spontaneous reaction. And so this is also why ATP gets used in biological processes, is it's perfect for coupling with these endergonic reactions. So these endergonic reactions, they have a positive delta G, they're non-spontaneous. So these would be some reactions that have to be coupled with these exergonic reactions as well. And so here's my coupled reaction, but here we're also seeing just this example of exergonic. So exergonic reactions, these would be my catabolic reactions. My endergonic reactions would be my anabolic reactions as well that have a positive delta G. We have to input that in there. So those would be some terms from energy to make sure that you guys are reviewing. Okay, now everybody take a big old breath because now we're going to talk about photosynthesis. So with photosynthesis, some things to make sure that we are addressing here is make sure we understand the overview of photosynthesis. We need to make sure photosynthesis first evolved in prokaryote organisms. We talk about cyanobacteria because remember that when Earth was formed, there was no oxygen in the atmosphere. And then when prokaryotes started using water as the electron donor, that released oxygen as a waste product. Well, that actually was a cause of lots and lots of death because oxygen was very toxic to a lot of prokaryotes at that time. So we'll make sure that we're understanding that, but mainly what we need to make sure that we are understanding is the light dependent versus the Calvin cycle and making sure that we're understanding some of the major events that happen with that and also making sure we're understanding where ATP synthase is located, where does the electron transport chain happen in photosynthesis and chloroplast is what we're going to make sure that we are addressing. So in this first diagram that I'm giving you, I'm making sure that you understand the overview equation for photosynthesis, carbon dioxide plus water, ultimately is going to lead to glucose and oxygen being produced, oxygen being the waste product, glucose being now the energy storage. This is an energy transfer process that is taking energy from photons and now storing that energy into organic molecules. Remember, with photosynthesis, we don't always just make glucose. So G3P is the main one that we need to make sure that we are recognizing. Now, a common mistake students make is they think, oh, cellular respiration is just the reverse process. Completely different biochemical pathway that utilizes oxygen, breaks down the energy from glucose in order to now create these waste products of water and carbon dioxide. This is simply showing us the cycle of molecules that cycle in between photosynthesis and respiration, but do not think that it's just an absolute reverse of the biochemical processes. There is no Rubisco in us. This is what I'm going to tell you. Now, if I look here at this particular picture down here, most of you would instantly look at this and think, oh, it's a chloroplast. No, it's not a chloroplast. It's cyanobacteria. So this is a prokaryote. And so this is just, again, reviewing and making sure you're recognizing that photosynthesis does not just happen in plants. There are protists that perform photosynthesis, and there are some prokaryotes that perform photosynthesis. So when we do talk about something like a cyanobacteria, do recognize that this is, being a prokaryote, this does not have membrane-bound organelles, but it does have membranes in here that can carry out electron transport chain as well. So this would be a prokaryote, and this would be the first prokaryote that performed photosynthesis. Okay, this should be very familiar to you. So this diagram we saw a lot. With this, we're trying to reference here what happens inside a chloroplast, inside of a plant cell. Now, inside a chloroplast in a plant cell, remember a plant cell is going to have hundreds of chloroplasts in there. Within there, these are my thylakoid membranes, and it's the thylakoid membranes that are responsible for the light reaction. And we'll take a look more in detail on the light reaction on the next slide. But I need you guys to understand the overview. It's during the light reaction that water is used as an electron donor. It's going to donate its electrons back to that chlorophyll A. The hydrogen is going to be carried through NADPH. Remember, NADPH is going to get reduced. And so this is an energy molecule. Oxygen then is the waste product that's happening here. And remember that light is captured in the thylakoid membrane through our different pigment molecules. And we'll talk more about photosystem 2 and photosystem 1 in capturing that light and transferring it to electrons for electron transport. Now, the light reaction's job is to convert energy from light into this stored chemical energy of ATP and NADPH. So the ATP that's produced here is through chemiosmosis, through ATP synthase. NADPH, this is now transporting electrons and hydrogen to the Calvin cycle, and it's these energy molecules that now in the Calvin cycle in the stroma of the chloroplast, remember the stroma is the liquidy goo filling that's here. This is where carbon fixation is going to occur, and this carbon fixation Now, through multiple processes that we don't need to know in a lot of detail, this energy is going to be inputted in order to now build larger organic molecules. And so these larger organic molecules, the output that cycles out of here is G3P, a 3-carbon molecule. So what we also have to remember is if I'm going to kick out a 3-carbon molecule, I have to take in 3 carbons in the form of carbon dioxide. Now, waste products of the Calvin cycle are now NADP+. So this NADPH it's going to drop off its electrons and hydrogens, and now it's NADP+, and it's going to go back to the light reaction and pick those up from photosystem 1. ADP and an inorganic phosphate are going to go back, and they're going to get recharged. Remember, I told you guys to think of these things as just rechargeable solar batteries. And it's the light reaction that is charging those up. Calvin cycle then is using that stored chemical energy in order to now create a more permanent stored chemical energy of organic molecules. So here's our details of the light reaction. What I'm looking at here is my thylakoid membrane. And in my thylakoid membrane, remember I have photosystem 2 is my first photosystem. My photosystem 2 is going to have a specialized chlorophyll A molecule that with this cluster of pigments that's embedded in here, light is going to be absorbed by the pigments, different wavelengths of light for whatever the pigments are, and they pass that to this reaction center. And at the reaction center, this is where an electron is going to be elevated to a higher energy state. And when that electron is elevated to that higher energy state, electrons now flow through these membrane proteins from higher energy state to lower energy state. And as they move, they're powering this proton pump to create a high concentration of protons inside the thylakoid space. Remember with basic diffusion, protons then, these want to move from a higher concentration area to a lower concentration area. So this is where I have my ATP synthase. This enzyme, remember it's this crank, this wheel, and as those protons flow through, this ATP synthase is now creating ATP. Now, remember that water is going to be used to replenish the electrons. So it's showing you here water being split, donating electrons. Oxygen then is a waste product that can diffuse out through the stomata. And then remember also that my electron, so this is my linear electron flow, my electron then can actually get re-energized by photosystem 1. Remember the wackiness of this. Photosystem 2 comes first, photosystem 1 comes second because they discovered photosystem 1 first. Now, photosystem 1 re-energizes that electron, and those electrons are actually going to get energized by this absorption of light, and it's going to end up going to NADPH. NADPH being a high energy molecule. So now that this has been reduced, now what can happen is NADPH and ATP can now be used by the Calvin cycle in order to convert carbon dioxide into larger organic molecules. So remember, this is all happening in the thylakoid space. Make sure that if you're ever asked where in a plant cell does electron transport take place or where is ATP synthase, make sure you're remembering that a plant cell has chloroplasts and it also has mitochondria. So we also have to make sure that we are remembering that connection. But if we're remembering this, electron transport generates the high proton concentration, the diffusion of protons through ATP synthase, that's my chemiosmosis. So we're going to see that again in our next section for cellular respiration. Now, Calvin cycle, when we talk about the Calvin cycle, you do not need to know any of these intermediate molecules. So that is told specifically to you by College Board that you do not need to know that. However, you should be familiar with this enzyme rubisco. Rubisco is the enzyme that is going to have a very high affinity to carbon dioxide and grab carbon dioxides in order to transfer them to this RUBP molecule that has to exist in order to now convert these inorganic carbons to organic carbons. This is called carbon fixation. Now, once I have these organic carbons, it requires energy in order to convert them. It takes energy and electrons and hydrogens from this NADPH in order to convert these to a G3P molecule. Remember, G3P is the 3-carbon organic molecule that can now be used for glucose or any other organic molecule. Once I have that G3P, So I had to have 3 carbon dioxides going in to get 1 G3P coming out. Well, now I have to have a regeneration phase that also requires energy in the form of ATP to get my organic molecules back to RUBP so the cycle can continue. So remember the Calvin cycle. Calvin cycle is happening in the stroma of the chloroplast. Carbon fixation is the first step. Then I've got my reduction. And so with this reduction, I've got this transfer of electrons as NADPH oxidizes. It is now reducing the organic molecule. So it's transferring the electron to the organic molecule to now create this G3P. So those would be some of the details of the Calvin cycle to know. These terms, if they ever use any of these terms of any of these intermediate molecules, they will give you this diagram and assume that you can read the diagram. Now, one other topic that we addressed, and this is just kind of one of those big idea number one when we talk about natural selection and evolution, we should understand that photosynthesis, there have been from C3 photosynthesis that we just talked about, where G3P, that 3-carbon molecule, is the first molecule produced. In dry, arid environments, plants evolve different mechanisms in order to prevent this water loss and also to prevent photorespiration. Because what happens, as we know, carbon dioxide has to diffuse into a plant through the stomata, but it's also through the stomata that water escapes. So in dry, arid environments, if water is escaping too fast, well, that closes the guard cells. And when you close the guard cells, well, now that lowers the carbon dioxide concentration. So what ends up happening is now oxygen gets grabbed by Rubisco and RUBP, which now is not going to lead to any development of any organic molecules. So if we're not developing organic molecules, there's no growth that the plant can actually have. So C4 and CAM plants evolved in order to combat this photorespiration in dry, arid environments. Now, when we have C4 photosynthesis actually uses completely different regions of the plant, different cell specialization. So what I end up having is I've got in the mesophyll cells, I've got carbon fixation happening with an organic acid happening here, but then transferring that organic acid to deep down into the plant, into the bundle sheath cells. And this is where the Calvin cycle is going to be taking place in a completely different region. So spatial arrangements are different there. When I have CAM, remember in CAM plants, now I just have a change in the night and day. What's happening? Because typically in a C3 plant, what's happening then is stomata are opened during the daytime when the sun is shining and photosynthesis can happen. Now, in CAM plants, the stomata close in the morning. So stomata are only open at nighttime is what's going to happen. So they're fixing carbon dioxide at nighttime, and then during the daytime, when now the light reaction can take place, that is when from the organic acids, they're releasing the carbon dioxide and Calvin cycle is taking place at that point. So those would just be some evolutionary changes in order to combat that water loss and also that photorespiration that happens in dry, arid environments. All right, you may want to pause and take a break and then come back to this topic of cellular respiration. Because with cellular respiration, if we look at the learning objectives and the essential knowledge, here's the first page. We talk about fermentation, cellular respiration. We are talking about electron transport chain, our NADH, FADH2. We're talking about a series of electron acceptors as they move through. We are talking about ATP synthase again and chemiosmosis. We are then also talking about glycolysis. We are talking about breakdown of pyruvate to as it moves into the mitochondria. We're going to talk about the Krebs cycle. We are going to talk then about all of this, which, again, you might want to take a break before you come back and understand more details, because photosynthesis was already enough to chew on. Now cellular respiration is a lot more. So if you have come back to me, or you're ready to just stick it out, let's just gut it out, Shelton. We got this. Here's the overview of cellular respiration. So remember, first of all, with glycolysis. Glycolysis is this energy release process that occurs in all living things. So this is another evidence of unity of all life on this planet, of how anything that uses stored chemical energy, which is just about everything, glucose gets broken down into two pyruvates. Remember, glucose is a 6-carbon molecule through a 10-step process that is all enzymatically controlled. 3 carbon molecules of pyruvate are produced in this process. This happens out in the cytosol, that liquidy goo of the cell itself. So there's no organelles that are required here. And in this process of breaking glucose into 2 pyruvates, so in this catabolic reaction, I'm producing 2 ATP molecules. These ATPs are produced through substrate level phosphorylation. So that substrate level meaning through direct enzyme interaction, through those collisions that are happening in the cytosol. And with this, because remember that this whole process here is a huge oxidation reduction reaction. Glucose, what's ended up happening is electrons are actually being lost from the organic molecules, and those electrons are being picked up by NADH. Remember NAD plus or FAD plus? These are molecules that can transport electrons and hydrogens to my oxidative phosphorylation event, which remember oxidative phosphorylation is electron transport, where oxygen is my final electron acceptor, and chemiosmosis, where this is my protons diffusing through ATP synthase. Now remember through all of these steps. So remember, all organisms perform glycolysis. But in eukaryotes, pyruvates will actually get transported across the 2 mitochondrial membranes. And in this process, remember, we're getting carbon dioxide as a waste product because pyruvates are going to be oxidized. Carbons and oxygens, remember I told you from glucose, it's the energy that's in the glucose that's the value. And it's the electrons that are in the glucose that are the value. The oxygens and the carbons are actually just going to be waste products that are just ditched in these as we go through. So from my pyruvates, my pyruvate is a 3-carbon molecule. Each pyruvate is going to lose a carbon and 2 oxygens. The electrons then that are remaining are going to get shuttled with NADH over to oxidative phosphorylation. I'm going to have acetyl coenzyme A is the final production. I had coenzyme A, and remember that each acetyl coenzyme A is going to now be carrying 2 carbons. There is no ATP produced in this process, but I am getting electrons that are going to be used later for this energy transfer during oxidative phosphorylation. Now, my 2 carbon molecules are then going to enter the citric acid cycle. The citric acid cycle takes place in the matrix of the mitochondria. The matrix is the middle area that's in here. And during the citric acid cycle, this is when the oxidation of my original carbons are going to finish. And so for each acetyl-CoA that enters the citric acid cycle, now I'm going to have 2 carbon dioxides that are released as waste products. And through this process, I'm going to end up with 2 ATPs per glucose molecule is what's going to be produced. So at this point, I have, through glycolysis, through pyruvate oxidation, through citric acid cycle, I have generated 4 ATPs per glucose. But what I've generated then are all of these NADH and FADH2 molecules that are now going to go to oxidative phosphorylation. This is what we really need to make sure that we're understanding is the details of electron transport and chemiosmosis, and collectively how this is described as oxidative phosphorylation, where we get the majority of our ATPs produced. Now, do also remember, so before we talk about that in detail, do remember that after glycolysis, because glycolysis is that universal biochemical process, and I have pyruvates, this is actually where cells will either perform fermentation or they are going to perform aerobic respiration. If oxygen is present, then pyruvate will get transported across the mitochondrial membranes, and then these steps will happen. And then if there is no oxygen present, then this is where fermentation occurs. And different cells are either going to have alcoholic fermentation or lactic acid fermentation. Our cells do lactic acid fermentation. And do recall that in the fermentation process, fermentation does not produce any more ATP molecules going from pyruvate to this end molecule. Remember, the purpose of fermentation was to utilize the NADH molecules, the electrons and the hydrogens in NADH. So now I had NAD plus that could now go back to glycolysis and keep and ensure glycolysis occurring. So remember the role of fermentation when it occurs. What we're going to be talking about, though, is making sure we're understanding after the citric acid cycle. Now we need to make sure that we're understanding this diagram. So this is a diagram that we saw quite a bit. And remember what we're looking at here. We are looking at the inner mitochondrial membrane. This is where electron transport chain happens. And this is also where ATP synthase is embedded in mitochondria. So remember, I've got the same ATP synthase in my chloroplasts as well. So my thylakoid membranes, and I'm also going to have them in the inner membrane. Out here is going to be my outer membrane. Now here's my NADH, here's my FADH2, and NADH then at this point is going to get oxidized, reducing these proteins. So remember, if you're reduced, you've gained an electron. If you're oxidized, you've lost your electron. And that's really what electron transport is oxidation reduction reactions. And those electrons are being passed from higher energy states on down to lower energy states. So as these electrons are moving through, they're actually powering these proton pumps, and proton pumps are actually pumping protons into this inner mitochondrial space, this intermembrane space between the inner mitochondrial membrane and the outer mitochondrial membrane. It's this high proton concentration that now, remember, charged particles can't diffuse through a lipid bilayer, but those charged particles can diffuse through ATP synthase. So as they diffuse through ATP synthase, remember, it's the crank, that moves ATP synthase in order to now power this production of ADP plus the inorganic phosphate into my ATP. So it's this flow of protons. I cannot have this flow of protons if I did not have this electron transport. Now, it is also through electron transport. When we talk about aerobic respiration, we have to know the role of oxygen in aerobic respiration. This is the only place you are going to be seeing oxygen being used. Remember, oxygen is the final electron acceptor of electron transport chain. So with this oxygen being here, oxygen is the lowest energy state for the electrons to move to. Forming water then is the waste product. If there is no oxygen here, then electrons don't flow. If electrons don't flow, then I don't have any NADH or FADH2. These are holding on to their passengers, and so I don't have any NAD plus or FAD plus in order to then run the rest of the processes that happen in the mitochondria. So everything shuts down if oxygen is not present. So do recall why we have to have oxygen, but with this too, let's remember where my high proton concentration builds up in my mitochondria, and then my chemiosmosis is this diffusion of protons through ATP synthase to generate ATP. This is my oxidative phosphorylation is what we're addressing here. Photophosphorylation is what we address then in photosynthesis. So please, please, please remember, do not fall subject to the common misconception. When we talk mitochondria and we talk chloroplast, electron transport chain happens in both, but we have ATP synthase embedded in thylakoid membrane in the chloroplast, the inner membrane of the mitochondria in the mitochondria. My proton concentration is going to build up in this inner membrane space in a mitochondria. It's going to build up in the thylakoid space in a chloroplast. So again, if you were asked about plant cells, plants have chloroplasts and they have mitochondria. So don't forget that. If we're talking about an animal cell or we're talking about a fungal cell, mitochondria only is what we are addressing in that particular case. Lastly, okay, now you guys can breathe because we got through some of the difficult stuff. So this last one really is just kind of understanding some fitness. When we talk about fitness, this is just connections between different molecules and changes in molecules in order to help the organism fit in their given environment. And so with this, there was some illustrative examples that College Board suggested that you are familiar with. So one of the things we talked about were different types of phospholipids in cell membranes. So in plants, remember that plants, when they have unsaturated fatty acids, these kinks actually allow for this cell membrane to remain more fluid as temperatures drop. So for the environment, if I am in a very cold environment, my assumption is that those plants actually have lots of unsaturated fatty acids in their lipid bilayers. That would be an illustrative example for understanding phospholipids. Now, hemoglobin is another one, and then chlorophylls is a third illustrative example. So let's talk about that. So hemoglobin, we did talk about the fact that your fetal hemoglobin and your adult hemoglobin is different. Adult hemoglobin actually has the alpha chain and the beta chain. So this gives me my quaternary structure of a protein in order to transport oxygen. My fetal hemoglobin actually uses an alpha chain and then a different chain. Well, what this does is this allows for a higher percent of oxygen saturation in the hemoglobin at lower pressures. So when you're in the womb, there is lower pressure that's in there. And so therefore, we need a higher affinity to oxygen for that fetus to be transporting oxygen throughout their body. So that would just be that change in structure in order to allow for that change in oxygen saturation, depending on the environment. So change in my protein structure gives different saturation rates of my hemoglobin depending on the environment. And lastly, you guys have seen this. So here's my absorption spectrum. What you're seeing here with this absorption spectrum, though, is you are seeing one, two, three, four, five different pigments. And so we can understand that depending on a plant and how many different pigment molecules they have, what wavelengths of light they can absorb. If they're in low light areas, then they're going to want to have as many different pigments as possible in order to make sure that they can absorb more wavelengths of light in order to generate more electron transport and more ATP and more NADPH. And if they're already in a high light, high intensity of photons, then maybe they don't need as many pigment molecules. So plants are going to have different combinations of pigments in order to be able to absorb different wavelengths of light. So again, that would be a change in environment. So there is Unit 3. So Unit 3, enzymes, understanding our energy concepts, photosynthesis, and lastly, cellular respiration. So review, review, review. I will make sure that I put up some practice FRQs for you guys. And so with that, then really just kind of see the different ways that College Board could ask you these concepts. But energy, very important theme, and guarantee you're going to be asked specifically about energy-related concepts with photosynthesis, respiration, and the energy relationship. So with that, as always, shoot me emails if you have any questions, and happy studying. Talk to you guys later.